the basal ganglia really isn't the only subcortical structure that is modulating the thalamus. It receives inputs from...
Is that there's a kind of a paleo-striato-pallidal circuit that is sort of the...
There's this fascinating, deep stories about, stimulating these diffusely projecting thalamic nuclei in primates that are anesthetized, and they wake up from anesthesia.
It's there in mammals, and it's there in birds, but it's unclear to what extent it's there in other animals and to what extent it's ancestral. So that closed loop action of the basal ganglia.
Welcome to Stimulating Brains.
Hello and welcome back to Stimulating Brains. So this episode is a bit special, and I had the great honor to talk to two heroes of mine in the same go. One is Associate Professor Mac Shine from Sydney University that we already heard in episode number 9. The other one is Associate Professor Paul Cisek from University in Montreal, who together bring so much knowledge and insight into the human brain to the table that essentially all I had to do is to lean back and let them exchange their thoughts of their brilliant minds. So we cover a lot of ground. What brought me into inviting Paul and Mac is that both have great thoughts about the basal ganglia, also about dopamine and how they interact, how the basal ganglia would modulate the thalamus to then speak back to the cortex and also, of course, to the brainstem. And we essentially delve across a lot of topics, even to some degree cover AI at some point, as a mistake of mine, but then also we go back 750 million years ago into evolution and talk of what Paul calls the phylogenetic refinement approach and how we can use that to better understand how the brain is made up by correlating the behavior that early ancestral animals could already carry out with the neural substrates they already had. And then if things change, especially topographical changes, happen during evolutions to these brains, if that was then correlated with new behavior that arises, then we can bring these together and essentially conclude that these new neural systems may have had an impact of these new behaviors developing. We also talk about how almost the entire forebrain, meaning the cerebral cortex and basal ganglia, mushroomed out of a part of the hypothalamus. And then we also go into Mac's ideas of how the thalamus integrates with the cortex and the basal ganglia and how they interplay. And if you're more interested in a deep dive of these ideas, you can give a listen to episode 9 again as a preparation to this episode. We'll try my best to add a few of the papers that were mentioned into the show notes on the website too. And we even have a guest quote from Haggai Bergman, another hero of mine, in here. Thanks for that, Haggai. And so I think a lot to look forward to, and I hope you like that. And I want to thank you wholeheartedly for tuning in. Spread the word about our little podcast. I think this episode could be more interesting to the neuroscience field at large. And yeah, thanks again, and have fun listening to Paul Cisek and Mac Shine. Thanks, Paul and Mac, for joining. Maybe we can add that it is evening for us on the East Coast, so in Montreal and Boston, and it is morning for Mac. And so we're here to talk about very exciting things today. And since Mac has been on the show before, I would pass the first 2 icebreaker questions to Paul, because usually before we get into science, I always ask about free time. So, Paul, what do you do when not involved in science? Any hobbies?
Well, I don't have a lot of free time, frankly. I mean, what non-science time I have is sort of spent treading water and trying to keep myself afloat, and help out around with the family and everything. But my wife is also a neuroscientist. So between the two of us, we're busy all the time and just scrambling. So essentially the free time is spent, hanging out with our son, doing stuff with him. That's become sort of the hobby. My hobbies are his hobbies. So, things like, watching, reading Harry Potter or playing Uno or Pokemon, or these are now my hobbies. It used to be things like playing the piano, which I still love to do, and he's doing it. So again, so it's kind of, Andrea also plays. So there's those things. But I have to say that when you asked me this question, I thought to myself, it's disturbing that I can't really think of anything because it's the time. But yeah, I mean, as a kid keeps you busy, as you know.
Exactly. Yes. Yeah. So I think that changed for me too. And it might still be, there might be a part of this being a disease under scientists that the longer you're in, it's just hard to maintain other things sometimes because but a kid is of course a great thing to spend the time.
Yeah, essentially I get to be a kid again, which is the main
yeah.
Absolutely. I get to play with Lego, which otherwise would be kind of strange if I was playing with Lego. But now I can do it, and it's totally acceptable.
It's great. So, yeah. Who have been key mentors in your career and turning points that brought you to where you are now?
Well, I mean, obviously my supervisors were a big deal. So I started as a computer science student, and I was all set essentially to become a computer programmer. I had, as an undergrad, a number of jobs, and then I was at Microsoft for about a year. And if I had stayed, I would have been quite wealthy.
Yeah,
but I did not. So I just decided I wanted to get into science. I was always drawn to science.
Do you regret it with Microsoft? Sorry to ask.
What's that?
Do you regret it not staying at Microsoft?
I regret not having the money, certainly. But because when I calculate out how much my stock options would have been worth now, it's depressing. But no, actually, I don't. Actually, I think this suits me a lot more. I don't think it's for everybody. I don't think scientific research is there's so many sacrifices and aggravations, but actually, it suits me very well. It's kind of fits my personality. So I kind of knew a long time that I wanted to do this. And then when I went to grad school, I went to the group of Steve Grossberg, and worked with him and Daniel Bullock. And really, I learned to think about the brain sort of through their eyes. And a couple of other professors there, Ennio Mingola, who sort of exposed me to ecological psychology and all of those ideas. So a lot of the groundwork was laid there. I think that's true for a lot of people that go to grad school. A lot of your worldview is set up by the people that you train with. And so, but then I went to Montreal to work with John Kalaska, who had kind of become a hero of mine because everything I read by him was great. He was always doing the experiment that somebody should do, in my opinion. And so then he was actually Steve Scott, who was here at the time, and John, who were my co-supervisors. They kind of taught me experimental neuroscience and kind of got me hooked on it. I didn't think that I would actually be running an experimental lab doing neural recordings in monkeys, but I really got excited about the quality of the data, just what you can do with that kind of data, which is not possible with other things, I think. Although, of course, there's some techniques that are even more impressive in mice and lamprey and such. But I got sort of hooked on the idea of having data, in part because I think it made me a better computational modeler, right? In Boston, I was just a computational modeler. Mm-hmm. But if you have data, then you can be a computational modeler who is not so deeply attached to the models, because if they're wrong, you still have the data, right? So you're not like your whole meaning in life is not tied up to some theory, right? The theory can be wrong, but then, maybe you'll come up with another thing. You've got data. You're still making a meaningful contribution. So, but in addition to that, having access to a lab actually makes it possible for me to do the experiments that I want to do, right?
Of course.
And it's harder to convince somebody else, to invest 5 years of their life doing some experiment to test my idea. But if it's me, then it's easier. So I kind of got hooked on that, and so really those ways of thinking. I mean, the the very heavily dynamical systems oriented way of thinking that Steve Grossberg pushed, for decades.
Yep.
That became my way of thinking. Ecological psychology became a sort of more philosophical grounding to all of it. A lot of the evolutionary stuff actually came from Dan Bullock and Steve Grossberg as well. And then actually doing experiments and actually delving into the brain and getting some humility about how complicated it really is. That came from John Scott.
And I assume running a monkey laboratory is a big deal, right? There's a lot of threshold that you need in terms of funding, in terms of facilities.
Well, the group here has a good critical mass. So there's sort of, because we have, we've always had about 5 people doing this kind of work, plus, others doing work with other animals. But there's enough critical mass that you're not really on your own. I think it'd be really hard to do it by yourself. But yes. I mean, I have to have a full-time technician, or I wouldn't be able to handle it. I have to have, good people always in my lab. And then I have to get grants to support all that. I mean, in the end, salaries are still the biggest part, but there's just all kinds of stuff, all kinds of complications, and you have to just sort of be on call, kind of, right? Because people need you. Now, again, once you've got good people, like right now, one student just graduated, but my other project is led by a postdoc. But he knows everything. He knows how to do everything. He can run the whole lab. So I don't really need to deal with every problem. But still, it's a daunting task.
Sounds great. Yeah, really cool. So we will talk about broader things, I guess, right? Zooming out, not so much about the hardcore electrophysiology data that you acquire. And Mac, I think next question to you. Many people have talked about this, including, I think, Bujaki and yourself and Paul. So, but just to set the stage for what we want to talk about, if you currently open a modern textbook of psychology and even sometimes of neuroscience, you will find terms such as perception, cognition, action, and subheadings such as attention, memory, thinking, and decision-making in it. And these terms actually read remarkably similar. I learned that from you guys. 2 terms used by early pioneers such as William James, or even by the ancient Greeks sometimes. And then a lot of these terms have not been created with the brain in mind, because at the time we didn't think so much about the brain. And so only later neuropsychology tried to match these terms with brain activity. How did that go? Was that successful? What are the challenges there? Can you talk a bit about that?
Yeah, it's a really fun question to think about and to really kind of marinate on just how difficult of a problem it is to understand how our kind of inner mental lives and our interactions with the world are related to brain structure and how you'd go about interrogating it. And we were actually just talking about this. I just got back from my lab's retreat for a few days, and in amongst them beating me at various sporting events and board games, we were talking about some of these problems, about how in some ways neuropsychology and cognitive neuroscience has really this difficult challenge on its hands, right? If you want to understand a behavior, let's say you care about something like an animal's ability to do some particular skill, enact a particular skill, grab something, to understand that in the laboratory, you need lots and lots of recordings of that particular thing. You can't just take one off and hope that you can somehow look in the sort of mass of all of their neural activity and understand what's going on. You need reproducibility. And so in order to do that, you need to design experiments that bring out that particular feature again and again and again. But without being, too hard about it, you're essentially kind of painting yourself into a bit of a behavioral corner in that way, right? You're forcing this really precise behavior on this sort of almost equipotential nervous system. And if you're not careful about it, in fact, I really learned this from watching some of Paul's early talks on YouTube when I was learning about his work. If we're not careful about the fact that we're building in this seriality into the kinds of challenges that we present to animals and humans in a experimental setting, if we then go and look at the neural activity and we forget that, we can sort of almost convince ourselves that what the brain is this kind of serial processing system. You think about it a little bit. Every time you go into a lab, if you've ever looked at a cognitive neuroscience experiment, oftentimes the challenge will be something like the following. There's a crosshair on a screen. You're then presented with some form of stimulus, which means that the first part of the brain that receives anything at all that's different from the crosshair is going to be the sensory system. You're then going to have to do some work to figure out what that sensory stimulus meant in the context of the experiment you're doing. And then you typically have to do some form of response. A motor response could be touching a button or moving your eyes to the side, saccading left or right. And so there's this sort of seriality baked into the way that we interrogate the nervous system, and for very good reason. But out in the world, it's much, much harder to find this kind of seriality. In fact, the world is much more parallel. While I'm driving my kids to school in the morning, I've got to take in visual information about what's on the road, what the cars are in front of me, whether there's a red light or an orange light or a green light. I've also got to be hearing my kids talk about their problem at school that day. Oh no, did they forget their lunch? Or what can I do about getting back to that? It's this massively parallel problem-solving challenge that I have. And it's really, really difficult to kind of imagine how you would investigate that in an experimental setting. So I think in a way, this is a really, really deep problem that we've got to figure out how to deal with. And I don't really have a great solution to it because most of the behaviors that we really deeply care about are these much more idiosyncratic one-off type of problems, at least in the lab at the moment. Paul, I'm really curious to hear your thoughts on how you tackle this as an experimentalist. I'm much more of a theoretician nowadays, I think, but
well, I have to admit that my experiments are actually still like that, so my theories are all, trying to address, how we actually behave in the world, like driving the car and listening to your kids and just how you deal with the constant changing flow of sensory information. And one of the most important things, I think, that people have recognized, of course, for a long time, is that it's actually closed loop, right? Your actions themselves are determining the next time series of sensory stimulation, and it's that actually that is the whole point of behavior, right? It's how to not crash the car and get it in the right direction, right? So that control aspect is part of it, right? But then the problem is that to understand it, you kind of want to do something a bit like engineering system identification, where you just sort of poke the system and see how it responds, and you poke it again and you see how it responds. And so that's the kind of stimulus response empirical method that we use. And I must admit that my experiments in the lab are extremely unnatural. They are these trial-based things where animals or humans do the same thing 100 times. And then we do a stimulus and we do a response, and then we repeat, an intertrial interval, and then we repeat, right? And now it's not necessary that the empirical approach becomes the theory, right? The theory can still be about feedback control and parallel systems, and it can still make predictions of what might happen if you were there to take that animal and stick it in that experimental lab. And you can still make predictions. So I think you can still address the question. If you have a theory, right, of how the system is dealing with the natural behavior, closed loop and all that, it might make predictions of what would happen if you took that closed loop control system and stuck it in a lab and gave it a stimulus over and over again. And what are some of the predictions? And so I think it still can be done. And there's a study that I found very inspiring to me, and it was a study by Anders Ledberg and others who put LFP recoding electrodes all over the brains of monkeys, and they showed them stimuli that they had to either let go at a level or not according to some rule, right? So they had a stimulus configuration, they had some rule in effect, and they had some response. So it's extremely impoverished, simplified task. But the cool thing about it is that the result is actually very pertinent to theories of how the brain works in parallel, because what they found is that there's this sort of very fast feed-forward sweep of sensory-like information, which discriminates things like categories, and it gets to almost the whole brain within like 50 milliseconds, right? Mm-hmm. But then the entire brain suddenly, almost simultaneously, about 100 milliseconds after that, discriminates and makes the decision, right? And you see that in parietal cortex and premotor cortex, and you see it in visual regions, actually some of the visual regions later than in premotor and parietal and such. So there's this kind of this kind of sweep of information, and then it gets colored by how the decision unfolds and across a very highly distributed network. And I was excited about this because this was fit very much in the kind of theories that I was working on with John Kalaska, about this kind of system for specifying the potential actions and then selecting between them, right? Now, in the context of natural behavior, you should be doing those things all the time, right? You have a system that's sort of a feedback system. It's appreciating the possibilities of actions while it's engaged in executing one of them, but at the same time, it's still sort of monitoring what other possibilities might be out there and sometimes switching. And that's kind of how we were describing this behavior. This is, of course, very much based on Gibson and others who came much earlier. But then you imagine, well, if you took that system and you stuck it in a lab and you gave it this task that they gave it, well, that's what you'd expect. You would expect this large feed forward sweep, because the information does have to travel in there, and then the whole thing sort of gets biased one way or the other. So I think you can do it. I think you can ask questions about natural behavior in a classical kind of experiment if you have a theory that actually makes a prediction, an interesting prediction for that kind of experiment. But not to go on it forever, but I do think what's exciting now is that people are starting to do closed loop experiments. A lot of interesting VR work done, a little bit in monkeys, a lot in humans, of course, but some of the best stuff is with mice on trackballs running through virtual environments, or zebrafish with stimuli projected into the tank, swimming freely and actually being able to sort of quantify the behavior, but still allow the animal to control the stimulus. So to control its full closed loop of interactions and actually really do justice to the richness of it.
I actually just heard an interesting experiment along these lines that you might find interesting, Paul.
Yeah.
There's a guy that I know at Macquarie University, Andrew Barron, who's working with David Kaplan. They're kind of embodied neuroscientists, an evolutionary neuroscience bent, and they've done one of these VR experiments, but they've done it in Drosophila, in flies. And so they have the flies that can, flap their wings either side, and then they have a virtual display that then dictates how the fly would move. So if it flapped its wings to the right, it should move to the left, and left to the right. And then they can switch it so that the animal, when it flaps its wings to the right, now moves to the right. And this is a little bit like this really, really fun video online of this. I think it's called Smarter Every Day, where this engineer in Alabama gets his friend to change his bicycle so that when he sets to the right, it goes to the left, and left to the right. And he can't do it, right? It's absolutely impossible for this man to do this thing. It takes him months and months and months of deliberate practice to learn how to do this thing. Really, really difficult. It turns out that
he can't ride a normal bike afterwards.
He can't ride a normal bike, exactly, right? So it's deeply problematic for him. It turns out that when they do this to the fly couldn't care less.
Oh, really?
Really?
Flaps to the right, moves to the right. It's like, so what? The world changed when I moved. Okay, big deal. Flaps to the left, moves to the left.
It learned.
No cerebellum.
It just didn't care. Yeah, and so their prediction is that because the fly has a much less developed cerebellum, there's less that's causing the problem for it that would be a problem for a vertebrate.
But wait, anyway, so it never adapts. It can never steer itself.
Yeah, exactly. It's used to being more pelagic, right? The wind whipped up and something moved, and it just kind of figured out the movement it needed to do in the moment. I just think that's a deeply fascinating example of some of the benefits and costs of having this big complicated nervous system, but it also, the kinds of things you can bring out with these closed loop type experiments. So I think to put a wrapper on your question, Andy, I think that if we can take very seriously the fact that the kinds of labels we've had in the past maybe were a little bit premature, and we start to look at the brain anew, kind of the Yuri Bajaki inside out idea, maybe some new labels will come up. Maybe some of them will be more about coupling with the world than about this particular function that you could put into a vat, like attention or cognition, and more about multitasking, the coupling at multiple different timescales or something. And we'll come up with a richer language, I think, of the kinds of capacities that we have that differentiate us from different species. In some ways more beneficial, in some ways worse. But it's a
Actually, I'd
fascinating.
like to add. I'd actually add to this topic because there's a kind of a historical point to be made here, and I guess you both have heard it, but just to say it. So psychology, the study of the human mind, had been separate from studies of physiology for a really long time, in part because there was this kind of idea that we're different, right? That we are special. We have a sort of some non-physical mind that is perhaps only humans have, and for hundreds of years, thousands of years, people have been thinking this way, right? And so psychology was actually psychology actually means the study of the non-physical mind, but nevertheless, even though it was assumed that this is non-physical, people were still said, well, we can still study it scientifically and systematically and very carefully. And people like, I'm blocking on the name, which is kind of embarrassing, but Wundt and Titchener, I'm thinking of. They did these studies very carefully, very systematic studies of how the mind works, right? And it was just a different topic, right? So at the same time, this was like in the late 1800s. At the same time, people were studying animal behavior. They're all talking about closed loop control, like people like von Exkull and others, and then later Tinbergen and Lorenz and all these people. Lots of people have been studying animals behaving in the wild, and all of that was really about closed loop control, right? Von Holst and Middlested very famously did exactly this kind of thing with the fly, except with much lower tech, right? They actually flipped the stimulus around, and all these kinds of interesting ideas had been there in the ethology and in the biology world, very much centered on this kind of ideas of interaction. But it was just not the same in psychology, and I think it's because of this idea that the mind is this separate thing, right?
Yeah.
Exactly. And now if you believe that the mind is separate, then you're sort of forced into believing there's some kind of interface between it and the world. So how it perceives and how it acts, right? And so I think what happens is that you have this assumption that forces these additional assumptions, right? And then the whole field, ideas like attention and memory and such, were all framed within the context of all this, right? And they got sort of solidified into very specific concepts that everybody knows what that is, what attention is, right? And what memory is. But then at some point, this idea of the non-physical mind just fell out of fashion, right? Largely because it was just not compatible with the laws of physics, right? It just didn't. At one point, people gave up on it, right? And it was behaviorists that really pushed that, sort of killed that idea, right? But then it needed to be replaced. And I think what happened is that the structure that was forced by that idea just stayed, right? Because we already had the terms of perception and attention and memory and such, and these ideas were already embedded in it, just the way we talk about it. But the whole time that psychologists were doing this, I think all of the ethologists and animal behavior people were going like, what is all that about? I mean, why are they insisting on these things?
People try to find the correlate, the anatomical or functional implementation of these concepts in the brain, right? But they didn't fit, I guess, because
we're still looking, and they fit a little bit. Right?
Sure,
there are some things like that, but I don't think they fit in the way one would expect. They don't fit in the way that maybe Titchener would have expected, right? Now he was a dualist, so maybe he wouldn't care, but I just don't think that the ideas, sort of the questions that we ask about how does X work, it has a definition of this thing called X that predates our study of the brain, as you said.
Yeah.
These ideas predate the study of the brain, and so. But if we had gone around about it very differently, we would have had different theories. I think we would have had much more focus on interaction, and James Gibson said all this, and many have said this. Piaget said all this too. So I just think they were always sort of, the kind of maverick outsiders. They weren't really mainstream psychology. So
Yeah.
so people didn't listen. Plus, Piaget was incomprehensible. That was also a problem. At least,
Yeah,
at
I
least.
heard that in your podcast episode with Paul Middlebrooks that you apparently try to read original and it's hard to understand.
I was actually reading in Polish, which was even harder. So I don't know why I did that. I should read some original Piaget, but I think I'd probably do better just reading somebody else's sort of concise,
Yeah.
translated version of it, because he had his own terminology. It's not easy.
So we mentioned Yuri Bozsaki, but and with his idea of starting from maybe the brain signals, so from inside out, to then come up with terms. I think another really promising concept, and not competing concept, that you've come up with, Paul, is to go back in evolution, right? So the human brain is the most complex object in the known universe. The three of us embarked on this impossible, maybe stupid mission to try to understand a bit of it. But one key strategy to maybe do so would be to study simpler brains. And of course, we can do that in animals, but we can also, we can go back in embryology. We might come to that, but we can also go back to animals in simpler forms and model organisms, and could use mice, but also could go back in evolution. And then if we thoroughly understand the brain of a model organism, such as the lamprey or the shark, which might be easier to understand, not as futile as a mission, we might then, of course, extrapolate some of these functions to our brain. And so based on this, or, not a new idea, of course, but you developed, I think, a really cool new framework, which you call the phylogenetic refinement approach, which adds the additional layer of behavior to this concept, essentially looking at which behavior was possible in these early animals and then which behavior came with new structures that formed. And I think by doing so, you came up with a preliminary but maybe more grounded tree of behavior in your papers that starts with simple things that simple animals could do already and then adds on to new things. Could you maybe give us an executive summary of the approach, how you came up with it, and what you learned so far?
Well, for me, it's just kind of been, I don't know. To me, evolution is kind of the grand unifying theory of biology, right? So yeah. It seems that we should use it, and the idea really came from a number of sources. I mean, lots of people have said this. And actually, my supervisors, Dan Bullock and Steve Grossberg, developed a nice model of spinal cord, essentially using this kind of approach. But people have said this very nicely before. The basic idea is just, we know that the thing was created by evolution, and evolution is actually quite conservative in terms of not throwing away old solutions, because you can't. If you have an escape circuit and you're living in a hostile world full of predators, you're not going to get rid of that escape circuit. There will never be a generation that doesn't have an escape circuit like that. It's just not going to happen. Well, I guess it could happen if you take a different strategy, like hide in the sand your whole life and just filter feed. What tunicates have done, essentially, devour your own nervous system and just build a hard shell. But in general, mobile animals and such are going to have certain constraints and how they're going to change at every particular stage. And so the point is this: if that's the case, then there should be a kind of a historical sequence of how those changes came about. And then the proposal is just, well, let's see if we can actually reconstruct what that sequence was. And people do, throughout biology, people do this. It's just a comparative approach.
Of course.
And you compare across species and you infer whether things are homologous, and then you know something about the common ancestor. I'm just saying, I have a term, phylogenetic refinement, but it's really the same thing that people have been doing in all fields of biology, except, cognitive neuroscience. So the idea is just to add a constraint on yourself when you think about mechanisms to make it necessary that whatever mechanism, whatever hypothesis you have about something works, has to be compatible with a stepwise process of how it could have come about. Now, the tricky thing there is if you're going from humans backwards, then it's very hard to know, because if I want to go back from humans and make my theory of humans consistent with a theory of monkeys, well, then I still first need a theory of monkeys, and for that I need a theory of some tree shrew-like creature, and then some common ancestor of mammals. So going backwards is very hard. Going forwards and doing it sort of chronologically is also hard, because we don't have those animals anymore, right? All the animals except the current ones are dead. So the best we can do is try to infer what those various ancestors were like. And for that, it's useful to look at specific species so you don't want to just look across all species, right? I mean, you can, but then you're still stuck with that first problem that I mentioned, right? If you look at Drosophila and octopi and birds and humans, good luck. They all are very, very, very complex, right? But if you instead, let's say, compare mice to lamprey, the reason why lamprey are really good is because they diverged from us, 500 million years ago, but then didn't really change that much, at least according to what we can see. From the fossil record, we can see something that looks like a modern lamprey 350 million years ago, and probably earlier. The Amphioxus is another good example. It's a creature it's a chordate that diverged from us about 650 million years ago, and didn't really change as far as we can tell, because you want to look at animals that didn't travel through too many different niches since they diverged. So Amphioxus and lamprey are great. Octopus, not really. The octopus diverged from us 800 or more million years ago, but the common answer was nothing like an octopus. I mean, it was nothing like an octopus or a human. They went through a crazy convoluted path that's super interesting, but it's not going to tell us as much about what our common answer was. Well, flatworms will probably tell us a lot more, right? Because they didn't change as much as octopi did. So birds are all, helpful, but confusing, and so on. So lamprey are good. Amphioxus is good. Sharks are pretty good, because they diverged. They became apex predators. And when you're an apex predator, you don't really want to change too much. And then you have a variety of other species along the way. And so I think by looking at those, you get an idea of what the various stages are. And then you can do what I'm essentially trying to do, is kind of try to constrain the problem from both ends. You have some idea of what the common ancestor was like by looking at these specific species to give you that.
How they behaved, right? Which
and how they behaved
did they have in their repertoire? Yeah.
Yeah, and of course that's a tough thing, because behavior doesn't directly fossilize, right?
Sure.
But traces of behavior do. You can get a lot of ideas from looking at the bodies of what animals were capable of and where they're found and things like that. So people do that. And that's actually the other thing I would say, that when I embarked on this, I was very encouraged by just how much people have done, how much there is out there that's been done, really good science on exactly these kinds of questions. So then you try to constrain it from both ends. You try to constrain knowing what the later creature is, and knowing what the earlier creature might have been. If you have sort of a Amphioxus and a lamprey that sort of bracket that particular stage, and you can say, all right, well, what happened? What might have changed? And I can tell you there, the telencephalon emerged, right? There was something like a hypothalamus, and then there was something that grew into a telencephalon, and that is interesting. And there's basal ganglia and all that kind of stuff in lamprey. So there was a big change there that happened. And another big change that happened is they became kind of predatory. I mean, they're parasites, but they seek their prey and they chase it down and stuff, whereas the Amphioxus is a filter feeder. So there's things that we can sort of surround with these convenient species.
So new types of behavior together with new brain regions.
Exactly. And what is it? Yeah, exactly. Like the Amphioxus doesn't really have an eye. It has a photosensitive patch that can't even make an image, right? But all it does is it sort of synchronizes its behavior to diurnal, like, cycles, circadian cycles, and hides from things. Every once in a while, it runs away from stuff, right? So it doesn't really have much of a visual system. Whereas the lamprey has image-forming eyes on the sides of its head, that allow it to guide itself through the world. And it has the circuitry that then supports that kind of behavior, including the telencephalon and such, and basal ganglia and all these kinds of things. So I think it gives us both, right? Because there's so many studies of the amphioxus and there's so many studies of the lamprey, that now you have a really good handle on some interesting events that happen along this path. And then you just try to find that for every little segment. And again, I'm encouraged by when I read this stuff and I see just how much people have done. There's experts on along the way that represent knowledge about each of those segments. Some of the segments are trickier than others, but there are people working on all of it. And essentially what I'm doing is just reading their stuff.
Really cool. And you write a book, right? I'm really looking forward to that. I think at this point, probably many listeners of this podcast will think, this is a bit of an odd episode, talking about amphioxus and sharks and so on. But what we'll get to what made me so interested in this work right now, essentially, because I think if you even go back more in time, 750 million years ago, the conceptual clade of eumetazoans roamed our oceans, and these were multicellular organisms already that had a nervous system. And I think you base this on work by Thomas Hills. They already used dopamine to switch between explore and exploit behaviors. And I felt, this is so cool that, first of all, I did not know dopamine already was in play at that time. And then, with your phylogenetic refinement approach, the idea of studying the behavior that was apparent in these smaller organisms already, could we potentially make the big leap through evolution to us humans and learn, about the role of dopamine better by doing so? And then maybe also we'll get to the basal ganglia in a bit. So maybe to ask Mac, do you have a clear understanding of what dopamine does in the brain? How would you summarize it? It's not an easy question, but
yeah. So I was just going to say thanks for the hospital pass, Andy. So yeah, the history of dopamine is fascinating, and I think it's also it's worth, while we're thinking about phylogeny, kind of admitting to ourselves that dopamine has sort of shown up and disappeared and then shown up again, a few times across phylogeny. In fact, it kind of fits really nicely into this idea of a sort of spare part that was sort of lying around from our diet. And if we need to, turn the volume up or down on this circuit in this particular context, or if there's adaptive benefit for that, well, the dopamine is one of these kinds of chemicals that's sort of lying around and can be used to kind of take advantage of that. So in terms of, the broader question of how to think about it, we place dopamine into this class of what we would call kind of neuromodulatory neurotransmitters. Their main mechanism of action cellularly is via G protein coupled receptors that have a different kind of an action than the kinds of neurotransmitters that affect ion channels directly. So if you're a neurotransmitter that opens an ion channel, you can enact a really, really quick change. You can cause, things like glutamate can cause a channel to open that causes sodium and potassium to flow in and out of a cell, change in action potential. You can also cause chloride, and you can kind of make a cell much more hyperpolarized or depolarized. This other class has a much slower effect, but it can be much more modulatory as well. So there are sort of two main classes of these G protein coupled receptors. Ones that liberate calcium, one of my favorite chemicals in the nervous system, and then another class that change things like voltage-gated ion channels, and the kind of, think of it sort of a little bit like the excitability of the cell, or the ability of the cell to respond to and enact change when it receives a spike. There's also a lot of really other fascinating stuff that happens on much longer time scales with plasticity with these different G protein coupled receptors that you don't find as obviously with the ionotropic receptors. in terms of kind of putting a wrapper on, what dopamine does in the vertebrate brain, it's a really tough one because I think a lot of it changes on context. I'm inspired a lot by the work of Joshua Burke. He has a really nice way of kind of framing this, where he thinks about dopamine as kind of important for kind of helping you elaborate decisions in an action selection, but also has this really important effect of causing plasticity, such that the things that you've done that worked well for you, or the things that you'd like to avoid, you can kind of reinforce those in the nervous system, such that the next time that you come back to that kind of same context, you know how to act in the future. And so it's got this kind of dual kind of nature to it, where it's helping you navigate the world in an adaptive way, but also helping you figure out how to do it better next time in this kind of longer time scale. So that's kind of my first protocol for dopamine. You often hear people talk about it as kind of, the sort of seeking system as well, which I kind of like as well. I don't like to think of it as kind of a pleasure chemical, which sometimes comes up in the kind of broader sort of non-scientific literature, just because that's such a much more complicated term that I think imbues a lot of other parts of the nervous system. But yeah, that's, the seeking system and reinforcing system is probably the best way for me to kind of start off there.
And so,
it's funny because I just read that paper.
Okay.
Sorry, I just read that paper by Josh Bork yesterday.
Oh, that's great, huh?
Rereading it because I thought maybe this would come up. So yeah.
So that's the good thing about Josh's work as well, just to kind of put a tap on that, is that he really is an empirical neurobiologist that does really beautiful work with rodents, where they'll go in with the tools of optogenetics and really brilliant recordings, but place animals into scenarios that really challenge some of our preconceived notions of these systems. And one of his works that I was really inspired by, refinement, was the first author. This is going back some years now. I met them at a Gordon conference in the basal ganglia. And what they were doing was placing these rodents in situations where they had to wait for a particular time for different levels of reward. And the way that they demonstrated how motivated they were for that reward was by leaving their nose in a little sort of part of the system, a little kind of, had to sort of stick their nose into a little hole in this apparatus, and the longer they left it, the more motivated they were for that reward. And they could actually show that the levels of dopamine in parts of their striatum were actually raising it and kind of co-varying with their amount of time that they would wait for this reward. But then what they also showed, which I think was really, really cool, was that if they then dropped the reward, if they then all of a sudden said, no, you don't get that trial on a set of catch trials, the amount of dopamine in the striatum would drop down. And if they looked at the difference in dopamine over time, right, if they took the step-by-step change in dopamine, it actually mimicked the kind of old reward prediction error story that was really popular back in the late 90s from Montague and colleagues. And so you could kind of have this idea of dopamine as this system kind of controlling your motivation, the amount in which you're willing to kind of engage resources to get a particular thing. But then if things changed quickly, either you got a big reward or a big drop in big punishment, now all of a sudden that was telling you about a prediction error that would then help you update your behavior next time. And I think the idea of finding these kinds of empirically driven descriptions of how a system could work can actually kind of really reinforce how we sort of think of the system as a whole. So I think it's a kind of nice example of what you were mentioning before.
So there's super hard evidence for everything you said. I think I totally agree with the reward system, reinforcement learning, also learning in that end, right? But one very big topic as well is Parkinson's disease, right? So why do people with Parkinson's not get depressed? I mean, they do sometimes, of course, but why is that not the predominant system? So what's the role in dopamine for movement?
Yeah, so I think the best way that I've been able to square this away with myself relates to the kind of differentiation that you see in the basal ganglia across the neuraxis. And the traditional story in Parkinson's is that you, particularly for akinetic rigid Parkinson's, is that you lose the dopaminergic innervation of the putamen, which is a more predominantly motor structure, before you would lose dopaminergic innervation of something like the ventral tegmental area to the nucleus accumbens. And so the idea would be that you could actually kind of reframe one of the issues that people with Parkinson's deal with as a lack of motivation for movement early on, right? They're less willing to engage resources in the detailed control of their movements over time. It's more effortful for them to create basic movements. And then over time, you start to see impairments as the pathological damage spreads to more anterior and ventral parts of the basal ganglia. You can have really difficult challenges with anhedonia, and other kinds of motivational issues at a kind of more behavioral scale later in Parkinson's.
Sure.
So that's
Later, I think things align very much with what you said before. In the earlier stages in the motor system, though, it's a great analogy to think of it as motivation for movement. Is there also a component of the reinforcement part of things in motor? Do you think? How do we bring the two things together? Also, a question to both of you.
Well, I think the motivational aspect's always there during the performance aspect of. So there's experiments and phenomena that sort of are pertinent to the performance of an action in the here and now, but that's potentially different than what you learn from the here and now, right? So what you learn for future movement. So I think dopamine has this motivating role, but it also has this learning role, as Mac was saying. So I think those are kind of different things. Now, I don't know whether the ability to learn from rewards or errors in Parkinson's appears at the same time as the kind of, as the thing spreads to these more anterior parts. Maybe that's the case. The putamen is probably more involved in learning action patterns and such. So I think I think the sort of deficit of motivation is a pretty good explanation actually for a lot of Parkinson's. It's the inability to sort of volitionally decide to go and do something, right? I mean, of course, we all know how excellent Parkinson's patients are in reacting to things that are really strong. If you draw a line on the ground, they can step over it, but if you tell them to just take a step forward, they have a hard time with it. If you throw them a ball, they'll catch it just fine. So there's a certain sense of it has to be sort of the internally generated thing. That's where the problems are more, right?
So, I mean, there's even symptoms like rigidity, which make it's hard to draw the line for motivation for rigid tonus of the muscles.
Yeah, so we're actually developing a model of this.
Okay.
And in the model, we suggest that the basal ganglia is sort of helping you commit, right? And so it's sort of taking information from the cortex and sort of helping the cortex choose one action among others. And if the actions are really clear, like you throw somebody a ball, then the cortex does not need basal ganglia to help out, right? But if things are ambiguous and you're not really sure what to do, then you kind of need the basal ganglia to help you select out from among all the things you can do, the one thing that you're going to do. And without that, it's almost like the classic ideas of Mink, that it's the inability to suppress all the other things that just you're kind of a deer caught in the headlights. You just can't go one way or the other way, and you're rigid in that sense. So I mean, it's actually kind of the old idea about basal ganglia, but I actually think it's so in our model we're essentially describing it in that way, and I think it explains certain kinds of data quite well. But one of the things that I always say about basal ganglia is that whatever theory you have, somebody has a piece of data out there that's going to, whatever theory of the basal ganglia you have, somebody else has got a piece of data there that's going to kind of cause trouble for your theory.
Yeah, I love that quote. That yeah.
And the thing is, I realize that one of the problems that we have is, as neuroscientists, is we always want the neural function of some structure to be sort of externally interpretable, right? Oh. But if the function of the basal ganglia is to change the balance of activity in the thalamus, I mean, this is something Mac has emphasized, right? If the function of the basal ganglia is to deal with the dynamics of the rest of the system, then until you know the dynamics of the rest of the system, you don't even have the language to describe the function of basal ganglia. And there's no guarantee that any particular one part of the brain can be sort of understood outside of the context of all the things that project to it and that it received from it, right? I mean, this is one of the things that's kind of daunting, that You know, we know what every neuron does, right? It contributes to the activity of the next neuron, right? But then, you know, you got to study the next neuron, right? And so that this is kind of, I think it's one of the problems. And I think the basal ganglia, for me at least, is one of these regions that I feel like, you know, any theory that I have that's sort of externally interpretable is probably wrong. I have to think about all the other pieces of the system that it's modulating. You know,
I mean, it is also, you know, probably involved in almost everything the brain does, right? So the striatum gets an efference copy of almost anything that's going on in the cortex. So it must have some major modulatory role. And then what always confuses me too is that most things work without the cortex, right? When there was no cortex, most of the behaviors we often study were already present. So then would the cortex just be an extra layer of compute there? You know, would the basal ganglia alone do the same job but poorer, you know, less elaborate and so on?
Well, this is where looking at evolution is actually quite interesting. So one of the things I've learned is that, you know, and you know, many people study the basal ganglia know this, that, you know, for a long time, the basal ganglia was the major output of the whole forebrain, right? It was not just the basal ganglia, but the sort of the extended Swanson-like basal ganglia, right? All those sort of striatal and pallidal-like subcortical ganglia. That was really the whole output, and the pallium, of which the cerebral cortex is one subregion, is kind of the front end to the striatum. Essentially, it's kind of the front end to the striatum, and the striatum then projects downstream. And in non-mammals, most of that is going down to the midbrain, and so it's modulating these midbrain circuits, and again, the striatum and pallidum are sort of the output. But it's modulatory, as Mac noted. You know, it's not driving so much as modulating the activity in the midbrain that actually governed a lot of behavior for a long time. And the input of the striatum is just that. It's just sort of the front end, right? And even the thalamic input, sensory input in things like fish and amphibians is a lot of it's going just to the striatum. It's not even going through the pallium, or, you know, the sort of cortical-like structures. So, you know, it's kind of like, you know, it's not subcortex, right? It's the cortex is the thin layer on top of that thing, right? It's not, you know, it's. I think when we study humans, we're very impressed with the cortex because so much, you know, that front end has become really elaborate.
Yeah.
But of course in mammals things change because the cortex does have its own projections out downstream and actually governing directly controlling things. But
so
for a long
pyramidal
time,
system essentially new thing, right? Came with mammals.
It's a bit of a debate. So a lot of people would say that, yeah, that's a new thing that mammals and birds came up with independently. That's what Streeter and Northcutt say, and they're real experts. But then again, Grillner, studying lamprey, shows that, well, there's descending output from the pallium in lamprey. So either lamprey came up on their own, or it's, as Grillner suggests, that is actually there. It's just it hasn't really become so massive until you get to mammals.
Makes sense. Okay, so maybe we can also mention in these very early cortical animals that had a like first cortex. I think the first regions that came up were S1, V1, and A1. So the primary sensory organs. I've recently read that M1 was not really present in the beginning, but was essentially still taken over by S1, like a sensory.
That's region. Still the case in marsupials. It's not really separate.
And then in between regions grew, right? So but these and in these animals, if you look at them, obviously the cortex is super small. It's just like another. You're right. Like looks like the brain looks very different, and then you have this small cap on top, right? That will become a big hemisphere, but it is not massive. It is not impressive yet, right? And it's still neocortex, I guess. Yeah. And so I think one of the, of course, key points you often bring up is I think you refer to the work of Puelis and have stated before that the entire neocortex and basal ganglia, so the pallium, grew out of the peduncular hypothalamus. And why is that? I think you have a theory of why that part had to grow and mushroom out so big versus the other part of the hypothalamus remained more or less as it was.
Yeah, so I mean, I am very, very strongly influenced by Puelas because I think his work is just so solid. And so he essentially lays down the anatomical foundations by looking at fate maps during development that cut across vertebrate species very nicely, even to the Amphioxus, actually. So he provides a kind of, he and the people that work, that do that kind of work, provide the kind of fundamental topology of the nervous system. But he himself doesn't like to speculate too much about function, at least not in articles. And so
he does in person, Paul, out of interest. I went to a 4-day masterclass of his, and it was just poetry. Like you'd go from this little detailed drawing with a bunch of black dots on a screen, and then he'd start talking about how you could play the piano while also having a conversation with your wife. And it was just so much fun to try to even imagine along with him what he was seeing, because he really is a brilliant scientist.
Yeah, actually, I got to know him because we did that book club on the
of course
book, and that was a lot of fun. So I mean, my stuff is kind of like, so a lot of my stuff is kind of, speculating on Puelas's stuff in ways that he might not want to do himself or might disagree with, right? But my take on this thing, so he suggests that there's this terminal hypothalamus that's sort of the top of the hierarchy, kind of. It's sort of the front end of the neural tube. And that includes things like the anterior pituitary and many structures involved in basic nutrient balance and things like that, and a lot of internal hormonal control. Whereas the peduncular hypothalamus is the thing that the telencephalon grew out of. And my take on it is simply that the peduncular hypothalamus is, so there's two kinds of sort of physiological control. There's the physiological control inside the body, which is things like temperature regulation and food resource allocation, etc., energy dealing. And then there's the peduncular hypothalamus that does the same thing, except that it's extending through the world, right? So temperature regulation involving things like going and walking and finding a shady spot, and food resource allocation in terms of finding food. And so that part of the hypothalamus expanded so much because that problem is such a complex problem, right? Once you deal with the external world, you have to sort of keep up with the complexity of that world, and it's just an eternally expanding problem, right? Because you can continue doing more and more sophisticated things in that complex world. So I think that's why that peduncular hypothalamus expanded into this whole thing that includes the cerebral cortex, hippocampus, basal ganglia, et cetera, amygdala, everything short of everything on top, outside of the midbrain, really. And I think it's because of that, because that problem is just so demanding. And again, there's always a value of expanding it further. It's
a never ending problem. You get better at modeling the world, but then you at some point you could maybe want to model the weather, and the problems keep up, right? With
well, because your competitors are also doing that, right?
Sure.
And your conspecifics are doing that too, and their complex behavior gives you a whole new domain of complex behavior to exploit and deal with, right? And you have to keep up with all of them, and they're keeping up with you. And of course that applies to completely different nervous systems too, like insects and mollusks and such. But the point is that the external world is just really demanding, Yeah, and it can expand to things like language and culture and civilization, even.
To get ever better at controlling, right? And controlling the environment. I think you also mentioned that the move to land added a lot of complexity to life, right? All of it.
Yeah, I mean, it was a very big challenge, but at first it didn't really add that much complexity to the brains. I mean, a lot of amphibian brains, even now, are not really much more sophisticated than fish brains.
True.
But there are certain things that happen when you get out on land, and one big one, which Malcolm McIver emphasizes nicely, and Barbara Finlay, is that vision becomes really useful once you get out of the water, because water doesn't really, you've got like 1,000-fold increase in visual range immediately, especially with larger eyeballs and small adaptations and so that now things become really sophisticated, and there's a great value in actually planning ahead. If you can only see, one body length ahead of you, you can't really plan that much ahead of you anyway. But if you can see, 600 meters away, that's a big deal. But there's a lot of challenges too, because obviously the bodies had to deal with, the locomotion is different, breathing is different. You name it. So there was a lot of challenges. But then I think what happened at first is the brains didn't really get that sophisticated. The visual system became more important. Amphibians became very good at navigating with distant landmarks because now they were there. But they didn't get really too sophisticated, except some of them, of course, became sort of fully terrestrial animals, amniotes, and then really took off into that world. But now it's a different world, right? It's kind of constraining in some ways more than the aquatic world, because it's really two-dimensional. Things are now obstacles, and so there's complexity that happens. So in addition to the increased visual range, there's just a new diverse world of opportunities to deal with and dangers to deal with. So a lot happened out there, a lot of modifications, and they seem to mostly have involved the forebrain. The midbrain circuits still became very sophisticated, certainly in reptiles and birds. But then the forebrain became much more diverse and kind of complex, right? So amphibians don't really have mortality-specific thalamic projections, as far as I know. So it's kind of a multimodal, mix of signals that goes to the forebrain. But once you get to amniotes, you start getting vision-specific regions and audition-specific regions and such.
Can I ask a question here, Andy?
Of course.
So my father, as an evolutionary biologist, and he loves to tease me as someone who studies the human nervous system a lot, that humans aren't particularly good at much if you compare them to the rest of the biomass on Earth. We can't jump as high as a frog can relative to its body weight. We're not as strong as an ant is. We can't fly at all. Our vision's pretty bad compared to an eagle. You can kind of like come up with all these different examples of things where humans are pretty bad. But one thing we are quite good at is being flexible. And I think that's kind of what you were pointing towards before, Paul, that if things change, we have this big, gigantic nervous system with so much complexity in it that we can kind of adapt to the changing niche. And one of the things that I've heard quite compellingly kind of brought to bear on why that adaptation is sort of allowed to occur in our brain's development is that we're neotenized, right? We have such a long time in utero, much more relatively than a lot of other species. And so we're born much less developed, much less useful, as it were. We kind of rely on our parents for, in Australia, it's kind of probably about 25 years or so after birth before we can kind of shit on our own. And so my curiosity is to you, Paul, are there other species that have kind of relatively stretched neonatal periods, and does that confer to behavioral flexibility? Like, does the New Caledonian crow, sit longer in an egg than another bird that maybe isn't as flexible?
That's a great question. Yeah, that's a great question. I don't know, actually. It would be probably pretty easy to look it up. We could probably Google Scholar it.
We should Google it. Yeah.
Yeah. That's a very interesting question, because I agree that is a big part of our specialty is this flexibility and social interactions and language and such, right? But all of that requires this great flexibility. And being sort of born early and sort of helpless is part of it. I don't know. That's a really good question, actually.
As a tangent, I also think we have in especially the newer parts of our cortex, we have less myelin than, for example, primates, and it might be a stretch, but I think there's people that have thought that this might be because more myelin is less flexible, right? More insulation between things. I don't know if that directly translates, but I've heard that. I think Bob Turner once said that in a workshop, that maybe that's an idea where these newer regions have to be more flexible than, let's say, M1, and so there's less myelin because less insulation and more flexibility.
Yeah, I mean, we keep getting myelination into our teens, right? I mean, we're not really fully wired up until we're in our teens, right?
Yeah.
My understanding of myelination, at least. But I mean, of course, we have a number of very particular expansion. If we want to talk about the transition from sort of basal primates to humans, it's a lot of expansion of cortex and the cerebellar regions that project to those expanding parts of cortex. So it's like those and the basal ganglia. So I think there is this kind of certain circuitry that expands dramatically. What I find interesting, though, is the topology is quite the same. I mean, if you actually look at the pattern of connections, it's actually quite similar,
Just added in between.
Yeah, just kind of like take the thing and just expand it forward. The whole idea of the prefrontal cortex sort of expanding out of premotor is pretty consistent with the anatomy. And if you look at different species like gorillas and gibbons and macaques that sort of try to get, so actually chimpanzees, I guess, would be better. You see this. So some people do these kinds of DTI studies and of different species, and they find essentially what one would expect if it really did expand in this way, and the cerebellum in parallel, right? And in fact, exactly in parallel with the expanding neocortical regions.
But that idea that you brought up, that the hypothalamus, that one part of the brain that dealt with the external world just kept growing, right? Because you could ever get better at controlling the environment. It's such a powerful one. At least for me, it was a really, really enlightening one. And
I should correct it. I should correct it to say, though, that all this time, though, it was, for a lot of that time, it was actually the midbrain that was actually controlling moment to moment behavior, right? The optic tectum. Yeah, and the tegmentum and MLR and these, mesencephalic locomotor region. And a lot of these regions were kind of running the show, and they were really running behavior, but they were running behavior in a relatively sort of spatially simple way. Yeah, and sort of like those systems guide you through the world, but the forebrain system, the part that expanded that peduncular hypothalamic system, the part that mushroomed, is the part that this sort of modulated that system about when to approach. Are you pointing at a good direction or are you pointing at a bad direction? And that is the problem, actually. It's almost like the guidance, the sort of visual motor, spatial motor guidance was actually relatively well established. Yeah. Very, very early vertebrates. And then it's the deciding what to do in what context and learning from cues and all those kinds of things. That's where the forebrain really took off, right?
So making these decisions in a way, adding much more compute to it, right? Adding more data, adding experience and so on. So one parallel that I find striking is with ChatGPT and large language models. The task we give them is very simple, right? It's really just predict the next word. But in order to become really good at that, you have to learn a lot about even the entire world. So I think in the first time we all heard about this, we just predict the next word. We didn't think this could, or I didn't think this could become anything sophisticated. But in order to become sophisticated, you had to essentially build a model of the world, right? And I think that's what these systems did too. So maybe as one question, if you really want to be really good at predicting the next word or the next thing that happens in the brain, in the world, do you essentially have to grow a brain, or do you have to grow something like that can model things? And what I find interesting in both domains is that the task is quite simple. The definition of the task is really simple. Control your environment, right? It's a simple recipe, but then to actually do it, you need a lot of stuff.
But I would disagree with that, though, actually, because I don't think ChatGPT is actually doing that problem, right? ChatGPT is not actually trying to control its environment. It's trying to predict the next word based on having passively absorbed a sort of a hierarchy of patterns of how words are structured in human communications, right? It doesn't have any access to the actual meaning that the words are referring to. And this is why I think I do not believe that ChatGPT has anything like our type of intelligence. It's just
I very much agree.
So,
sorry. Just to clarify,
it's essentially ELIZA, right?
It's a very different question. One is controlling the world, the other one is predicting the next word. But the analogy I see is that it is a very complex task, and if you just add at it and try to ever come better at that task, you need to develop a complex system. It's a very different complex system. I totally agree. It's not a brain, right? It doesn't have consciousness and so on. So, but it still had to. You could easily, create a system that predicts the next word just based on some rough statistics, and it would, be better than chance. But I sometimes feel like it has to have a model of some understanding, if you will, so to be as good as it is. Maybe you disagree.
But
I don't. Okay.
But it's very easy. It's very easy to have ChatGPT say really stupid things. I mean, it's right. I mean,
Paul. Yeah.
I think what it is doing is what you said, that it could be just picking up the statistics of the actual language that's out there. And I think, that's exactly what it's doing. And I think once you start testing whether it's doing that or true understanding, then it shows its limitations. It's this kind of out of distribution thing and problem. And it's really, I think, fundamentally limited by this, the fact that it's actually just being trained. It's just being trained to reproduce the structure of human conversations, but it has no access.
Are we more than being trained?
Absolutely. How? Because when you learn the meaning of, and here I'm going to just sort of describe the Piaget view, right? When a child is there in that crib and he doesn't know what on earth is going on, he's going to, through trial and error, try a bunch of different things. And some of the things will work really well. And one of the things that works really well is if your parent happens to be in the room, then just making some noise will solve whatever problem you have. You don't have to communicate to your parent, my diaper needs changing. You just need to make some noise. The parent's going to figure it out, right? It essentially, think about it this way, is that babies have at their disposal this incredibly complicated and sophisticated animal that's incredibly easy to control. It's like this hopeless, helpless slave that will do whatever you want, whether it requires just, bringing some milk or, driving to a hospital in a high-speed vehicle, and not getting into a car crash. So the thing is, babies have this incredible resource, right? This parent, right? And they first learn to control the world through the parent, through a variety of different noises, right? But at a certain time, and the parent will happily do all that. And then at some point, the baby and the parent will actually help the baby to make some distinctions between the noises it makes and the actions that happen. So that if you really want food, you make one type of noise. And if you really want water, you make a different type of noise. And the parent will actually help the child make these distinctions through its own actions, right? And then you gradually learn how to control your parent better and better and better. And that's, I think, the essence of communication. So in other words, the meaningful interaction of satisfying your hungry tummy via the parent, that's the fundamental thing, right? And along the way, there's this very brief little, and that's a very complicated interaction. It's a very complicated dynamical system that involves the parent going to the refrigerator and all that kind of stuff. But along the way, within that control loop, there's one little thing, which is this utterance that's being made. And that works because the parent does the rest of the work, right? And so those little snippets of of cues that we send to our parents to get what we need to help them, to sort of, that's, I think, the essence of communications, this persuasion aspect of communication, right? And then it just explodes from that, right? Because the parents are willing and helpful to teach you all kinds of more sophisticated layers and layers above that. And I don't want to belittle the complexity, but I don't think at any stage it really becomes anything other than this kind of extension of control into more and more complex domains, like then it becomes a question of establishing your social status by interacting with people in ways that benefit you in the long term, because then they're your allies in case things go certain ways. And so, and I think those interactions are fundamental, and the actual noises made between the agents are just little cues we send to ourselves, and they're structured in a certain way because the interactions are structured, right? But ChatGPT and all these kinds of things are never given access to the actual meaningful things. They're just it's like, you could have a model of an airplane that predicts the noise made by the airplane, right? You could probably decode what altitude it's flying at, or whether it's landing, or whether the gears are down. You could make a model that predicts the sound of the airplane and does it extremely well to every level of detail that you'd want, right? But it's not the airplane. It's just the sounds the airplane makes. So I think what we have.
It would also do terribly if you tried to fly the airplane underwater somewhere you'd never recorded sound.
Yeah, that's true. If you go out of distribution, out of distribution.
Well, it extrapolates beautifully. Sorry, guys, it does, right? So I mean, there are even if you. I think there was this great example that I saw on Twitter where somebody asked, okay, if you put a thread of wire through a loop and then go back the same way out, is the wire now connected to the loop? And then the answer was no and was correct. And the other way would be if you go through the loop and then around the loop and then back, is it now connected? And it got that, right? So,
but is it extrapolating, or is that out there?
It's hard to imagine that it just read that somewhere, right? It's not just repeating what the internet says. It has to form a model, some sort of model. And it's not a human brain, I totally agree, but there has to be a model of creating some sort of understanding, quote unquote.
I'm not convinced. I think
me neither. I've done
many other examples.
I've asked it to stack blocks. Yeah. I've asked it to stack blocks, and it's it's very impressive. Don't get me wrong, but it makes mistakes that humans would never ever make, right?
Which humans, right? It depends. Also, think of, I would say humans make tons of stupid mistakes too, right?
Yeah,
so if you look at the entire population, so and even us, of course. So I very much agree. It's not like the human brain. I guess the point I wanted to make, and this conversation is not even about ChatGPT, of course, is just the idea that if you just hammer a single problem long enough and hard enough and give it as much resources as you want, with essentially being able to grow a brain, will it potentially lead to some sort of model of the world, right? And with the idea, if you would ask it, let's say you ask ChatGPT version 6 that will come up, and it knows you, Paul, it knows you, Mac. What is meaning for Paul? It would probably give a good answer. And it might.
I've asked it questions that are pretty my own research, just as a thing. And no, it didn't get it right. My research is not out there enough. I think if I asked it, actually, what it did is it ascribed it all to James Gibson, which is actually probably pretty fair, right? But
it's better than most humans would answer it.
Yeah. Well, anyway, I actually wanted to say that Alison Gopnik had a nice summary of this. She sees ChatGPT and a lot of these things essentially more akin to a library than to a human, right? It has an enormous repository of knowledge and a way of sorting through that knowledge that lets you get at it quickly. But and of course it's very different because it's creative in a certain sense, right? But it's not doing the same thing. It's a little bit more like a search engine that gives you responses in a textual form that combines information very nicely. But I really think I think that it's not because you can't do it, but I think what we've asked it to do is very far from what we're doing.
I think you mentioned that control, like meaning is lacking, right, for these systems.
Yeah, I think that's the thing. I think the meaning is lacking because meaning comes from I think it comes from the interaction. This is, again, very Gibsonian. It comes from closing the loop and understanding how things along the loop are actually purposeful in very basic things, like that terminal hypothalamus. It's all about keeping the terminal hypothalamus happy, right? That's the hierarchy. That's the part that says I'm well fed, I'm not I'm not being eaten, and I'm comfy, and everything else is just keeping that thing happy and essentially maintaining your structure. It's autopoiesis, right? Maturana and Varela. Those are the foundations of living systems. And then the point is that along the way, we come up with very convoluted control systems. And within that, the elements of those control systems have meaning because they are actually coupled to that outcome, that desirable outcome, right? The thing that's tricky is how those things become explicit and communicatable, where we actually have, because you can imagine, a control system spinning around and driving a self-driving car through the world or whatever, and it's very sort of pragmatically, dynamically coupled. But then the question is how we can then sort of come up with a more detached understanding. So Giovanni Pizzullo calls this a symbol detachment problem. And I think that's the right way of asking the question. So there's this question of symbol grounding, which maybe you've heard from Stephen Harnad. How do our symbols get attached to the meaning? The symbols being like words that we use. But I think I agree with Giovanni that the problem goes the other way. We have interacting systems, and the question is, how do symbols arise within those systems and become sort of detached from that dynamical context? And I think in the case of language, it's because, again, the interaction between parent and child has inherently in it a symbolic step, right? The noise that the baby makes that says, I want food or I want water, is symbolic because you don't have to tell the parent where the food is, or how to place the feet and how to hold the bottle. The parent takes care of all of that. And all you have to do is just send a little sort of a very symbolic and compact cue, but it's enough. And I think our language becomes symbolic because our behavior is because the behavior of the units in the world that you're sort of controlling through is so sophisticated and categorical in some sense, right? You either approach you can tell somebody to back off. You don't have to tell them exactly how to, again, place their feet. It's the control system is accomplished simply by making the utterance. And so
my grad students have the same strategy, Paul, when trying to go to international conferences.
What's that?
My grad students have the same response, the same strategy when they try to go to international conferences. I want to go. They don't know how hard it is to get the funding to go.
Oh, I see. Yeah. Well, if you're a good parent, you should take care of all that for them.
I do.
The details of all that. You should book their flights for them and find them a nice hotel.
So there's a nice analogy that we talk about a bit with the students here that might be kind of helpful, which is this notion that I think kind of comes back to the idea of control that you've been thinking a lot about, Paul, which is just how different a control architecture would be if you had more elaborate control to kind of enact on that control process. So if you think back to like a simple worm, you can think about the control that it has over its environment as something a little bit like a joystick. It can kind of like move forward and backwards or left and right. It could kind of copulate or defecate or something like that. And if you can imagine that as a simple joystick. Now, if you sort of elaborate a nervous system, now you've got much more connections between the input and output structures. Now, all of a sudden, you've got a little bit more like a sort of car where you can kind of imagine putting indicators on. I intend to move to the right so that anyone behind me can now see that I'll move to the right before I actually do the movement. This is very, very useful if you're trying to have social communication. And if you have a more and more elaborate architecture, now it's a little bit like being in a sort of advanced cockpit of a plane. Now you've got all these different gauges and dials you can move around. I can tweak the pressure here. I can kind of change the system. I can move this engine to that engine. I can pull full force or less force on the engine. And you can imagine that having more and more elaborate control systems can give you more and more elaborate kinds of ways of responding, some of which will be social, like you're talking about, and other ones will be more that multitasking we were talking about driving in the car and listening to the children at the same time. So I think this is a kind of a nice way to think about one of the things that's sort of a benefit of a more elaborate nervous system over phylogeny is this kind of more elaborate control architecture. Anyway, this is an analogy.
Yeah, do you know Tony Prescott's work? He's done a lot of interesting theoretical work on layered control architectures. But Bill Powers did this too in his classic book, and Ashby, and they a lot of them are actually using. They were using actually these metaphors. How does the pilot fly the plane like in dense fog, and again, it's the control of the setting of the dials that he's doing. He or she is doing, keeping it within particular things, like, for example, the altitude being above zero, but also other things, right? And so it's the idea of a controlled variable that you want to keep within a certain range, and then you have various ways of affecting it with all kinds of complicated dials and switches. But then you layer on top of that a thing that now is going to set the range that the lower level system deals with, and enact control through the dynamics of the lower level system by knowing its controlled variables, right? And this is almost certainly what happened, right? I mean, you have a spinal cord that you can steer by sort of differential activation of the mesencephalic locomotor region, which then generates oscillatory activity in your fish-like swimming thing. And now you can steer by this, and now you can have another system built on top of that, et cetera, et cetera. And that's almost certainly what happened. But one of the things that's hard for us, for neuroscientists, of course, is that, again, it's hard to interpret the stuff that's built on top of old stuff until you know the dynamics of the old stuff, right? And its particular intrinsic properties that might not be easily, describable even, right? You need to have a really good computational model before you even know what the control signals should look like.
Yeah, totally agree.
Yeah,
and we talked basal ganglia a bit more. So I've talked I told Hagai Beckman about our conversation, and he's another hero of mine beyond the two of you. And he has a guest question to you guys, and asks, and he's, by the way, I think both a fan of your works, Paul, but also your recent thalamus work that you've published in Progress Neurobiology that we talked about last episode, Mac, as well. So he asks, why are the basal ganglia the only subcortical structures that modulate the thalamus? And I think he himself, I know that from his conversation with him, has shifted a bit, being one of the typical basal ganglia model people to, since recently, believing that they mainly modulate the thalamus and the cortex. So I think he's been recording in the thalamus a bit. So that's why he's so curious about this modulation aspect there.
Yeah, I mean,
I think it's a good one for Mac.
Yeah, well, I want to also reflect here just a little bit of meta. Hagai is an absolute hero of mine, and I remember reading his reinforcement and dimensionality reduction model back in my PhD and just being blown away by how wide-ranging and thoughtful that work was. It was hugely inspirational to me, and making sense of some of the really difficult clinical problems like freezing of gait that we were trying to think about at the time. So to be asked a question by a guy is a deeply warming moment in my career. So the question, I think, is a great one. I think we think a lot about the thalamus as a really useful kind of starting place. If you're going to try to kind of grapple with some of those nice questions that Paul was asking before about how to think about different parts of the nervous system as this big interconnected network, The thalamus is a pretty decent place to start. The hypothalamus is also a really excellent place, but the thalamus itself is interconnected with pretty much every other major system we can think about in the brain. And it has this really fascinating architecture that kind of sits in amongst all of them. But I think it's really important to note that the basal ganglia really isn't the only subcortical structure that is modulating the thalamus. It receives inputs from the cerebellum, and often different parts of the thalamus will receive inputs from the cerebellum. The superior colliculus can impact the thalamus. In fact, superficial superior colliculus projects to different parts of the thalamus than the deep superior colliculus. Same with the inferior colliculus. There's a massive modulatory input from the brainstem, the adrenergic system, the cholinergic system, the serotonergic system all impact the thalamus deeply, histamine. And so I really think of it more, rather than being as kind of the sort of downstream gated sort of system of the basal ganglia, rather think of it as kind of the central processing hub that receives lots of different kinds of inputs and then has a way of helping to coordinate them. Now, I don't really love this analogy of the conductor because I don't like to think of homunculi and a kind of infinite regress in the nervous system, but it's kind of conductor-y in the sense that anything that goes on in the rest of the brain is going to have a little bit of an impact on this system, which then has this really interesting kind of inhibitory global modulation from the reticular nucleus. It's going to kind of shape and constrain what else can be active. And so we think about it kind of as this kind of really crucial processing hub in the center of the brain. Paul, I don't know if you have thoughts or anything you'd add to that.
Well, I would have. Yeah, I mean, I think I'm glad you answered first, because there's not much more for me to add. I mean, I guess I didn't quite maybe I didn't quite understand what the question was getting at, but I mean, I guess I would add that the basal ganglia projections to the thalamus are what I focus on, etc. But of course they're not the only ones. I mean, the basal ganglia is actually modulating many other things as well, and so, you know, I don't know if the answer is the same for the other things it modulates.
To go back to the cortex, the main route is the thalamus, right? So
yes, to get back to the cortex, yeah, it's the thalamus. Yeah. But there's a lot of downstream stuff, and here's something that Mac and I have actually talked and scratched our heads on about a bunch of times, is that that closed loop through the thalamus back to the cortex is it's there in mammals and it's there in birds, but it's unclear to what extent it's there in other animals and to what extent it's ancestral. So that closed loop action of the basal ganglia projecting through the thalamus, essentially modulating the thalamocortical system, that may be rather mammal specific. Now, I don't actually know which way the balance of evidence goes, but if that's the case, then that's really a big deal. That's a really big change in the mammalian circuitry. Whereas in most other vertebrate species, the projections are mostly downstream to like the substantia nigra reticulata, right? So the GPI and the substantia nigra reticulata are not similar structures in many ways. They're, in Puellas's model, they're completely different segments of the nervous system. And I sort of think of the substantia nigra to be sort of the output it's sort of the basal ganglia target of the midbrain, whereas the GPI is kind of the same thing, but for the forebrain, right? And it's sort of like that, through that loops of the thalamus, it's coming back and controlling and modulating, you know, the activity in the forebrain. But the projections from striatum down to and GPE to SNR are actually quite different, and they're modulating those midbrain systems.
That's very interesting,
and that's very old.
So and is that one reason why there's not really a homologous region of the GPI? There's a homologous region of the GPI in mice, but it's not called GPI yet. So is that the reason that these things were new and came in, like this loop back?
I think I don't know. Again, this is one of those things that I'd really like to get some expert to tell me what just the truth is, but I think maybe there's no agreement yet. But I know Ann Butler said this, and I think to Mac directly, correct me if I'm wrong.
Yeah, that's right. We had a chat.
Yeah.
chapter.
She believes that's a mammalian innovation, and that's a kind of a big deal, because before then, and it's, to me, it's interesting that we don't know whether these things happen hand in hand, but in mammals, what you get is a lot more descending control from the cerebral cortex, and at the same time, you get this loop. And that's the same case for birds. Birds have very strong projections from the forebrain downstream, and they also have this loop. Whereas in reptiles, it's kind of debated. Now, I don't know. I sort of go with the Grillner story that's actually ancestral. But even Grillner, I don't believe he's identified the loop back through thalamus up to pallium and in the lamprey. So that loop might be a new thing, and that's kind of important, I think.
Very, very interesting. And
speaking of collaborators that work on zebrafish, the thalamic architecture is really, and tactile kind of interactions with thalamus is completely different than the kinds of things that you'd read about in rodents and primates. So I think there's kind of good reason to think that there's quite a lot of important variability in this kind of topology that could be really important, as you say, Paul, to really kind of focus our attention on if we're trying to understand interspecies differences. But there's
I always get very excited about topological differences, right? But because regional size differences, it's less, I think, less telling, right? It's the topological differences can really eliminate certain theories, favor others.
And then that loop back up from GPI to thalamus back to the cortex. I think in your work, Mac, you based this on Ted Jones' work to think that the basal ganglia are mainly project to matrix thalamus neurons, and they would rather project diffusely to the apical layers, right? And the diffuse term is really important here to me, right? Yeah. So because I think we often learn that it goes back to the same side where it originated, and hence it's a loop, but it seems to be much diffuser. And I think when we talked in Montreal, Paul mentioned it's even in streaks that go. So can you talk about that, both of you?
So I was actually recently, after our conversation, I went back and reread another paper I loved in my grad studies by Nambu, the 6 problems on the basal ganglia. I don't know if either of you have read this, but it's this really lovely paper that kind of talks about where the field is at in the basal ganglia and kind of what we need to figure out as a group. And in fact, in that paper, Nambu brings up the fact that the diffusely projecting thalamic nuclei were actually discovered back when I was about 4 years old in 1986, but were placed in this kind of oddities basket. Like, we don't quite know what this is. We know that if you look at the palatal projections down, we stain these particular cells from the globus pallidus, the cells in the ventral tier of the thalamus. Particularly, I think in this case, they were looking at ventral anterior thalamus, project back up in this really diffuse fashion, often to supragranular layers, often hitting multiple different sort of functional regions of the cortex. But it was the sort of functional importance of this was I think really difficult to really pin your finger on. And I think once you then take that more global perspective and think, what is it that this circuit could do in the context of the rest of the system, I think it becomes a little bit more clear, at least. I don't think we know for sure right now, and I think that the modeling work suggests that there's a benefit to this kind of a structure, but the empirical work is still being done. But one of the benefits you could imagine is that if a cortical region is currently projecting down to the striatum, doing something a little bit like what Paul was suggesting before, kind of determining whether or not one of 2 options is the better one to go. Let's say you're in a 2 alternative force choice, deciding whether to reach right or left. You're currently arbitrating between whether or not you should do one of those 2 different things. The extent to which you can liberate some of the diffusely projecting thalamus to come back up to the cortex, innervate the supragranular regions. What you're allowing now is the feedback projections from higher cortical areas to have an influence. Maybe the context reminds you of something that you saw before, or you think, oh, I've seen 4 lefts in a row, maybe it'll be left again. Or, remember that last time when this didn't work, we had to switch. You're getting these higher order kind of more meta level concepts coming in that can then shift the balance of your decision so that you can change that action. And like I said, this is very theoretical, but you can imagine now that having that variability baked into the decision-making process is incredibly important for adaptive behavior. You don't just want to do the same thing every single time. You don't want to become stereotyped. You want to be able to use the rich context that you have and all the information at hand to make an adaptive decision. And so in our work, we've been trying to think carefully about the benefits of that. And, the reason that we're so excited by it is that if you go looking in the literature, there's this fascinating, deep stories about, stimulating these diffusely projecting thalamic nuclei in primates that are anesthetized, and they wake up from anesthesia. I mean, this is a deeply important set of structures, these diffusely projecting thalamic nuclei, that are linked to phenomenal consciousness in a really, really fascinating way. And so we're trying to think about how we can model them, how we can think of the computational benefits and costs of this kind of a system. And it's really early days, though. I don't think we're.
But what you're saying is that this one mechanism that you hypothesized about would be that if a cortical region is essentially in the process of deciding it could, by this loop, recruit more other cortex diffusely to propagate back. Is that, did I understand that correctly? So essentially,
yeah. So the way we're thinking about it now, if you dive in and have a look at some of the dopamine receptors that are expressed heavily on the direct and indirect pathway in the basal ganglia and the striatum, the classic story is something like D1 receptors are expressed on the spiny projection neurons that represent the GO pathway. This is something like projecting down to globus pallidus internus, which is then gating the thalamus. These are excitatory modulatory receptors. They're called GS, so they're going to basically make it a little bit easier for that cell to fire. On the indirect pathway, you have GI receptors. These are on the D2 type cells. So these are the chemical here will now cause a decrease in excitability. They're going to hyperpolarize the cell, make it harder for it to spike. It turns out if you track up into the cortex and look at the pyramidal neurons that project down to these different populations, it turns out that the pyramidal neurons that project to each of these two different populations also express these different receptors as well. And so the layer 5 IT cells that project to the GO pathway are expressing dopamine 1 receptors. They're going to be excited by the presence of dopamine. And the pyramidal tract neurons, these are layer 5B neurons that typically project to the indirect pathway, are actually expressing predominantly D2. So now you've got this now more elaborate role for dopamine to play, where this arbitration process can be gated by the presence or absence of this motivating chemical. So now, rather than just wondering about whether or not I should open up the channel in the cortex, I'm also wondering about whether or not I should vote for a different process, let's say, of the different options in the cortex. And here the thalamus can now open up the different options that you could be arbitrating. So we think about it as a really big emergent process shaped and constrained by dopamine that's playing out over the neuraxis, right? Not just focused down in one population, but rather across an elaborate kind of set of neural circuits. And here the diffuse projections can essentially buy you that variability you need in order to arbitrate correctly in a really complex situation that you might find yourself.
Very cool.
Yeah.
A different way of simplifying brains, and I briefly talked about it, is embryology, right? We can go back into developing brains and look at them. And I'm a big fan of the Swanson work and his a concept of looking at these earlier developed brains where you see specific folds that essentially divide the cortex from the striatum from the pallidum. And then we've talked about it briefly. Some of these pallidal sides would become the actual pallidum, but they would also become other brain nuclei. So I think in this Swansonian way of thinking, almost every single brain nucleus would be either a striatum or a pallidum. And so, first of all, do you agree with that? And then second, how would that fold into the general concept of the basal ganglia? So is his canonical circuit still valid in humans? So yeah. Go ahead.
Well, it agrees very nicely with what Puelas is doing and the fate mapping studies, right? So there's a essentially Puelas's model has the same subdivision that Swanson does for the forebrain. Now, I should make a caveat that these two groups actually argue a lot against each other. So there's a lot of disagreement, and I can get into that. It just has to do with the sort of the organization of the tube. I favor Puelas's view, but what happens in the forebrain in Puelas's model, that peduncular hypothalamus expands into a telencephalon, which has those three segments, as you said, a pallial segment, which includes things like the cerebral cortex, a striatal segment, which includes things like the striatum plus other things, like parts of the amygdala that are actually striatal, and a palatal segment that includes the globus pallidus, but also other things like, again, parts of the amygdala. Actually, the bed nucleus of the stria terminalis is sort of the pallidum for the amygdalar circuit. And so in Puellas's model, and this is, I think, fairly well established now from multiple labs, is that there's a kind of a paleo-striato-pallidal circuit that is sort of the basic organization of the forebrain. And then there's multiple circuits. So we all know the basal ganglia circuits, so the limbic, the motor, etc. But that can be extended into a hippocampal circuit, which includes the hippocampus and other parts of what's called the medial pallium. That's the pallial part. Then there's the striatal part, which is the lateral septum, and a pallidal part, which is the medial septum. And they're organized sort of topologically, kind of like the basal ganglia. And then there's the amygdala that has basolateral amygdala, sort of pallial. Then there's the central amygdala, which is sort of striatal. And then the bed nucleus of the stria terminalis, which is sort of pallidal. And so there's this kind of beautiful conserved architecture in parallel through all these systems. And the basal ganglia is one of them, and it's a very big one. But it's actually one of multiple cortico-striato-pallidal loops.
Would they all go back to the thalamus, by the way? Do you know that?
No, as far as I know, no. I think the dorsal striatum one does, the ventral striatum one does too. The amygdala, I believe so, but I'd have to track down the references for that.
And there's a lot of
actually, tell you the truth, if I were to default, my default assumption would be yes. Actually, they do all go through the thalamus, but I wouldn't be very confident about it. I mean, obviously, the basal ganglia one does.
Well, if what we said is true, that what we thought is true, that this invention is a new one, where the pallidum was actually project back to the thalamus, it could be that was an invention just of that system
that
and not of the other ones.
Part. Yes. But I don't know, to tell you the truth. I believe Loretta Medina. I mean, I heard somebody else say. So someone else told me that she said, and she would know a lot about this stuff, that the ventral does project back. I can tell you that Steve Wise and Betsy Murray in their books suggest that they all do. That structure is conserved. There's a thalamic, there's a pallial part, striatal part, pallidal part, and a thalamic part, and all of them go back. And again, these are people that know a lot about this stuff. So
sure.
So, but I don't know exactly, because there seems to be some disagreement to what degree it's all there or not there. But the organization of these domains from a developmental standpoint seems to be very well established in all vertebrates, actually. So there's a medial pallium in fish, which happens to wind up on the lateral side of the brain, that has that same topology as ours, our hippocampal circuit. So again, that I'm simplifying this a lot, of course, but there is, I do agree that with what you said, that Swanson's basic framework, I think, is fairly well supported. That part of it's very well supported by Puellas's work, even though they disagree vehemently on a bunch of things, as well as by other studies, based on connections.
When you read these things, you really get maybe there is some, basic Bau plan and some grand unifying principle that we might at some point really understand and, understand.
Yeah, but how much of that is just our wishful thinking?
Yeah, I know. And I think a lot of exceptions develop along the way, right? That's the other point where I think, again, it is not as simple anymore as in the lamprey. It is not as simple anymore as in a baby or a fetus that develops. So, yeah.
But there's a few people that have managed to unravel it a bit. I mean, I think Swanson's work for me was also very important, and Puellas's, he combined that. But another one I would add is Helen Barbas, right, who has the structural model that you may be familiar with. The basic idea that there's this concentric ring organization to the forebrain, which many people have said for a long time. But she suggests how that maps to the actual projection patterns that you get from sort of primary sensory regions, which are sort of central, that sort of central island of that concentric ring, and that how that projects outward in both feed forward and feedback ways. And I describe that as kind of an antidote, a sort of very welcome antidote to the Van Essen-Fellman diagrams, which are just like, here's the brain, look, and it's just like this incredible plate of spaghetti. It's like, oh my God, we're never going to make sense out of this, right? But in Helen Barbas's hands, it actually unfolds into something which has a very beautiful and relatively simple structure. Now, I don't know, maybe it's just her wishful thinking and mine wishful thinking, but I find it quite compelling, right? And then the nice thing about that is it agrees, I think, conveniently with a lot of very theoretical ideas, like predictive processing ideas of Friston. Lisa Feldman Barrett, I think, has summarized this link very nicely, sort of combining Friston's predictive coding and Barbas's structural model into a nice architecture that might be common across many species of mammals, certainly, and then actually kind of a very good overview of the anatomy and function of these systems.
I'll certainly check that out. Thanks for pointing that out.
Well, I mean, I just find it nice.
Yeah.
Because I get really depressed when I look at the real anatomy. When people show me the connectome,
yes,
God,
that's usually my emails with Haggai Beckman too, where I feel like I might have understand a small thing, and then he sends me a paper that crushes it and has tons of exceptions to the rule and so on.
Yeah, but if there is a rule, then I can handle the exceptions, right?
Yeah, you're right.
If it's just exceptions, then I just can't assimilate, and then I just give up and do something else for a living, right? So I kind of feel like we need that.
I think I want to be mindful of your time. I promised 2 hours only, and I don't want to go over time without asking first. So usually we have wrap-up questions with rapid-fire questions, but maybe just one for each of you. Same question: what's the future of neuroscience gonna look like?
An easy one.
Yeah.
So I just recently was quite lucky to be able to work on a paper with Emily Finn and Russ Poldrak about what we kind of saw the future of human neuroimaging, what role it could play in neuroscience, and it was funny. Our discussions kind of were quite polarizing. They were either deeply pessimistic or extremely optimistic, and the pessimistic version is so far we haven't done a particularly good job of connecting together as a field. Neuroscience is really young in a lot of ways, and deep insights in the cellular world or the rodent literature or the cerebellar literature or the human cognitive neuroscience literature are often just completely at odds with one another. But I think there's actually a reason for optimism in the sense that we're now at the place where we've got so much data and these amazing tools, things like dynamical systems, computational modeling, that we can start to try to figure out whether we can knit them together. And so writing this article, we had the students in mind. We thought, if you're a student coming along and you want to help out, the trick to me is try to learn from a few different fields and try to see if you can figure out ways to knit things together. Go read some brilliant old people like the Steve Grossbergs of the world and try to see if you can map it up with the kinds of neuroimaging you can do nowadays. And it's a hard problem, and it's going to, I think, have a lot of false starts in it because of how difficult it is to talk across communities, or to, as Paul was saying before, to take these ideas of dualism, which were based on this old idea of, psychology being separable from neuroscience, and then find a way back to those beautiful concepts, but from the neurobiology. But we've got an opportunity to do that. And so to me, that's an aspirationally exciting and optimistic viewpoint, which is that we are separated as a field, but we can integrate, and we should be thinking very, very long and hard about how to do that better. If we can, neuroscience will start to be viewed, I think, like other really, really tight interested medical fields, cardiology and renal physiology and things of that nature, where we can really make some inroads into disorders. So I'm optimistic for the future.
Paul, what do you think?
I'm fairly optimistic about some things, but I find some things very frustrating. I mean, I think one of the wonderful things we have to look forward to is new data and new techniques and this, just the size of the data. But one of the things that I find very frustrating is that I'll be kind of crude about this. It seems to me that there's some kind of a limited resource in science in general, where the bigger the data, the smaller the theory, right? It's almost like when we have more data, we're less prepared to do something with it that's actually hypothesis driven, right? And so a lot of research becomes very descriptive because it just has so much data. It's so far beyond any particular theory that a lot of people are doing very wide recordings, right? And then what they do with that data is they decode something, right? And because there's tools for doing that, it's easy to say I can decode this from that, right? But what is the theory that motivates that process? Why would you want to decode some variable X from some large data set? And I find what is actually the theory? It's not really a theory in terms of mechanisms. It's just kind of the presence of information. And I think almost every theory that you could conceive would suggest that there's the presence of information, there's correlations there, right? And I don't think those analyses help us distinguish between those different mechanisms. I really like dynamical systems that are actually structured sort of to actually express a hypothesis, such as there's on-center off-surround interactions in the visual system that creates edge enhancement or certain kinds of things, and then that does something, and then we can look for and we can make predictions very specific, right? But if you just decode the information is there, then the only hypothesis you're testing really is that the brain is responsible for behavior, which kind of obvious, right? And so what I'm seeing is that as the data gets bigger, I don't know, and I think the limited resource that I mentioned is just time, right? That a researcher really only has so many hours in the day, no matter how hard you work. And if you spend all of your time dealing with the massive data, you just don't, you get overwhelmed by it. And I think I see this happening to me as well, but my data is not that big yet. So I feel there's a bit of a, right now, a kind of a frustrating trend away from really hypothesis-driven research, right? Just because you have more data doesn't mean you can actually test hypotheses better. An example I really like is the sort of the test of the Newtonian versus Einsteinian model of the universe. It doesn't matter how many observations you make. Most of them are not going to distinguish between those two models. There's really only a couple of observations that are actually even pertinent to testing between those hypotheses. So if you collect more and more and more and more data, astronomical data, it's not necessarily going to help you. You really have to focus on the particular things where the models make different predictions, right? And again, massive amounts of, if I recorded from every neuron in the brain at high resolution, I wouldn't know what to do with it. I just wouldn't know what to do with it. But if I have a hypothesis about what some region will do in some behavioral context, then I don't need the whole brain. I just need really good data, not big data, but good data from those particular conditions that tell me about different mechanisms and from the particular structures that my hypothesis says I should be looking at. And so I think this kind of more targeted approach really works well in science, right? There's an old article by John Platt called Strong Inference, which says certain fields of science make really good progress, and it has to do with how much they are really hypothesis driven. I know, call me old-fashioned.
No, but I mean, we just had Marcel Mesulam visiting the Brigham here, and he said exactly the same thing. We need more theory, right?
We need theory.
I have a quote from John Platt on my desk, Paul. I love that paper.
Yeah. It's a great paper. And I think one of the problems we have is that we love the techniques. I would actually like to have recordings from every neuron in the brain.
It'd be great.
But I would not know what to do with it. You know,
We'd figure it out. Come on.
well, no, but I think what happens is people are taking the first step.
Yes.
No one has time to take the second step because the first step is just so gigantic. You know, maybe what I would do is I would throw away 99% of the data and just ignore it, not let it distract me. But then of course I feel like that's stupid. Why throw away all this stuff that's potentially insightful? So I think there's a I don't know, maybe it's just because the data has grown so fast, right? That the theory's not keeping up with it. But I think there's ways of, again, I think the key really is, have a hypothesis and maybe express the hypothesis actually as a computational model. And it doesn't have to be precise. The good quote from John Tukey is, far better to have a vague answer to the right question than a precise answer to the wrong one, right? And I think if we make models that are trying to capture principles and make distinguishable predictions, we're much better off than making models that describe every single neuron's firing very precisely, but don't actually, we can't even interpret the model because it's just a gigantic neural network that just fits the data, right? I think I find it frustrating that I see a lot of great data, and then I'm kind of like, well, tell me what it means, don't just describe it with a decoding analysis or representational similarity analysis. I mean, there's something to be learned there, but that next step has to be taken, in my opinion. We'll see what happens to me when I get more data.
Well, the answers I think will end up being deeply multidisciplinary as well. I think we keep coming back to this idea that if we want to be really good theoretical neuroscientists in my group, what really matters is that we collaborate with an extensive group of people with different backgrounds and different insights so that they can say, hey, you're looking at the fMRI data like this, but the electrophysiology data doesn't look like that at all. Or you're taking this dynamical systems approach. What if you took a cognitive neuroscience approach? You'd be missing this entire thing that we've been spending all this time clarifying. And so to us, the real key is finding ways to communicate to a broad group of people from a different set of backgrounds to try to find those lingua francas, right? Those little examples that they can all connect to, and then you can understand one another. And that's hard work. But if we can get that, then I think we'll be way ahead of where we are now.
Yeah, I agree. And I think that's actually some reason for optimism, because there's a lot more of that now. There's a lot more actually collaboration between people with different expertise and actually managing sometimes to understand each other. Yeah, I think that's good. And that's the other thing that I think that is important, is actually have a few people that speak that actually know enough about both fields to sort of provide that glue, right? Yeah. That's tricky. That's tricky, but if you have enough of them, then we can all glue together.
Make some progress. Yeah.
Yeah, I agree.
Thanks so much for your time, and this was super cool. So thanks a lot for participating, guys.